Hi, year 11s and 12s. We're going to be looking at our second video in the PowerPoint of cell respiration, which is Unit 8.2, a higher level topic for biology in your IB diploma. As I mentioned in the last video, we're not going to have the need to know in this video. They will be available in the OneDrive on the PowerPoint if you'd like to look at those. But let's begin. Last video we looked at the basically how to draw the mitochondria. We also looked at the different types of processes that we're going to need to know for aerobic respiration. So, like I said, glycolysis is the very first step in the controlled breakdown of carbohydrates, and it occurs in the cytoplasm of the cell. So this is part of aerobic respiration. It's also part of anaerobic respiration, so both processes use glycolysis. At the end of glycolysis, the pyruvate molecules that are created will look at the different types of environments they're in and will decide if they're going to go through anaerobic or aerobic respiration. So to start with glycolysis, we have our hexose sugar, which is a sugar with 6 carbons. So if I write 6C, that means the sugar has 6 carbons. It's broken down into 2 molecules of pyruvate, which have 3 carbons each. So you can see we've gone from 1 6-carbon to 2 molecules of pyruvate, which is 2 molecules of 3 carbon. So it still adds up to 6. We can summarize glycolysis with 4 different events: phosphorylation, lysis, oxidation, and ATP formation. Okay. PLOA is a good way of remembering that. PLOA, P-L-O-A, phosphorylation, lysis, oxidation, ATP formation. So phosphorylation, this is when the glucose is phosphorylated by 2 molecules of ATP. So 2 molecules of ATP are each losing their third phosphate molecule, and they're adding them to the glucose molecule, which forms hexose biphosphate. So our hexose sugar and 2 phosphate molecules. So 1 phosphate molecule from each molecule of ATP added to the glucose to create that. That phosphorylation makes glucose, or now hexose biphosphate, less stable and more reactive, and it also prevents it from diffusing out of the cell because it's too big. Okay. We go through lysis. So we've got our hexose biphosphate, which is still a 6-carbon molecule. It's split into 2 triose phosphates. So hexose 6, triose 3, and we've got 2 of them, each with 3 carbons. We've still got our 6 carbons. So gone from 1 hexose biphosphate into 2 triose phosphates. We then go through oxidation, where we've got our electron carriers. So hydrogen atoms are removed from each triose phosphate through oxidation. Oxidation is loss, and that reduction is gain, NAD+, to form NADH, and also 2 hydrogen molecules. Okay, 2 molecules of NADH are produced. So hydrogen atoms removed through oxidation, then those hydrogen ions, because oxidation is loss, are used in reduction. Reduction is gain, to create NADH, and 2 molecules are produced. We then go into ATP formation. So some of the energy that's released from these sugar compounds in the last 3 steps is used to physically make ATP. And we call this synthesis of ATP substrate level phosphorylation. In total, through all of these processes, and through substrate level phosphorylation, we create 4 molecules of ATP. But we say glycolysis produces 2 molecules of ATP. That's because even though we're creating 4 here, we had to start by using 2 molecules of ATP to make glucose more reactive and be able to break down. So we've actually used 2 molecules of ATP to get the 4, so we've really only made 2 molecules of ATP at the end of this. We've also, at the end of glycolysis, we've ended up with 2 molecules of pyruvate. Okay. We've also ended up with 2 hydrogen carriers, which have been reduced via oxidation, so we've got 2 lots of NADH and hydrogen ions. And like I said before, we've got our 2 net molecules of ATP. So what happens next is we have to determine what's going to happen with our new molecules of pyruvate. Okay. So glycolysis itself is an anaerobic process that occurs in the cytoplasm of the cell, as I said. So this means we're happening without oxygen, and we're producing 2 molecules of ATP. Now, depending on the availability of oxygen where glycolysis has occurred, this will determine what we're going to go into next. Are we going to go to aerobic respiration or anaerobic respiration? If we go through aerobic respiration, we're going to be we're going to be breaking down pyruvate in the presence of oxygen, and we're going to create 34 molecules of ATP. So the aerobic respiration creates 36 molecules of ATP in total. Whereas if we go through anaerobic respiration or fermentation, we're breaking down pyruvate in the absence of oxygen, and we're not going to create any further ATP, and anaerobic respiration is only going to physically create 2 molecules of ATP, which was the ones produced in glycolysis. Now, in aerobic respiration, we're going to look at this in a lot more detail. If oxygen is present, the pyruvate will be transported to the mitochondria for further breakdown, so that's complete oxidation. And this further oxidation is going to make large numbers of reduced hydrogen carriers, NADH or FADH2. And in the presence of oxygen, these reduced hydrogen carriers are going to release all their stored energy to create more ATP. Now, aerobic respiration is going to involve 3 more processes, which we'll look at in further videos, which is the link reaction, Krebs cycle, and the electron transport chain. Now, if we were to go down anaerobic respiration or fermentation, like I said before, we have no oxygen. So the pyruvate is not broken down any further, and no more ATP is produced. So we're at incomplete oxidation at this point. The pyruvate will remain in the cytosol, the cytoplasm of the cell, and it'll be converted into lactic acid in animals, or ethanol and carbon dioxide in plants and yeast. Okay, these conversions are reversible, and that's necessary so we can ensure that glycolysis can continue to create its 2 molecules of ATP, and we're continuing to get ATP in the body, or the organism. So what's going to happen is glycolysis. We've had the oxidation and reduction reactions that created our hydrogen carriers, creating NADH and H+. Typically, those hydrogen carriers are going to be oxidized via aerobic respiration to create NAD+, but in the absence of oxygen, glycolysis will quickly deplete any available stocks of NAD+, so we're not going to have any more, and that's going to stop glycolysis from occurring further. So because we've had no NAD+, that's the limiting agent here. Then fermentation will occur, which involves a reaction that oxidizes NADH back to NAD+ to restore those available stocks. So in basically we're summarizing that in aerobic respiration, we're constantly producing NAD+, but in anaerobic respiration, because we don't have that NAD+ being replenished, we run out of NAD+, which means glycolysis can't go further, so pyruvate has to go through a reaction that oxidizes NADH back into NAD+, so glycolysis can happen again and create those further 2 molecules of ATP.